Paul Thomas, Senior Editor of Pharmaceutical Manufacturing Magazine and PharmaManufacturing.com. And I'm here at Interphex 2009 with Ian Sellick, the Director of Marketing for Pall Corporation. Ian, thanks for being with us today.
Thanks very much, Paul.
And Paul usually makes a splash at Interphex, and this year is no different, especially in the area of disposable technologies. One of the things you've got going that's new is your Stax depth filter cartridges. Tell us a little bit about Stax.
Yeah, sure. Well, obviously, single-use technologies have becoming more and more used in the industry. Everybody's looking for ways to make the process safer and easier and to have less involvement with complicated equipment that needs a lot of cleaning. But one of the areas that has not really been addressed up until now has been the large-scale separation of cells and dealing with the first clarification stage of the process. So what we have done is to engineer a very simple, easy single-use system based on modular devices that allow you to separate cells or any other types of solids in pharmaceutical processing. And because people are familiar with the types of depth filters that we manufacture, what we wanted to do was to provide an encapsulated version of those so that people didn't need to revalidate their entire process to adopt this type of equipment. So inside each of these capsules is exactly the same media in exactly the same configuration as people are used to using. So there's no change in flow geometry, there won't be any change in performance, no change in materials of construction, so that the revalidation question becomes one of only saying, okay, we have an external shell which is additional to the process that's replacing stainless steel, but that in itself is exactly the same materials as people are routinely using in their process. Mm-hmm. So what we tried to achieve with this was a simple system that required minimal requalification, but equally because depth filtration is dealing with very large volumes of very messy stuff. If you can imagine a 10,000 litre bioreactor and the amount of cells and stuff that come out of that, the previous processes have actually been quite challenging in terms of dealing with the mess, actually, once you took the modules out of a stainless steel housing, which was the previous way of doing it. And some of these housings were very large. They required cranes to actually take the tops off the housing, and often the last thing you want in a clean area is a crane hoisting bits of stainless steel around. So when we were designing this equipment, we tried to make it have the minimum amount of hardware involved, but to make each module of a size that a single operator could quite comfortably actually manipulate it, and to make it so that all of the modules just literally clip together, so you can actually develop a process as big or as small as you want using identical modules.
So simplicity is obviously one of the benefits. What else are your customers telling you in terms of the benefits that they're finding?
Well, the other benefit is that by actually having a simple system like this, the whole system is actually a lot smaller, and it's small to the extent that, in point of fact, as you can see with this, it's on casters. Instead of having a very large piece of equipment which has to stay in place and have its own dedicated part of the facility allocated to it, now you can save a lot of space by actually just wheeling this in when you need it in the process, and so. You can wheel it in, do this separation, nowadays typically into bags or biocontainers, such as the totes and biocontainers that we provide. You can wheel those in. Once this is done, you just wheel it back out of the way, and maybe the next stage of the process, maybe a chromatographic process, you can just wheel in a different piece of single-use equipment to handle that as well. So the modern manufacturing plant is becoming much more flexible because of that. You can now just basically bring in equipment for each of the unit operations actually as you need it, rather than having a huge suite of permanently affixed equipment occupying very, very expensive real estate, and just bring in whatever you need for whatever stage of the process you're at.
And speaking of flexibility and single-use equipment, you've also done some new things with the Allegro biocontainers. Tell us more about those.
Yeah, that's right. The Allegro biocontainer range is a range of biocontainers where Paul has used its expertise in polymer chemistry and knowledge of polymer films to manufacture what most people colloquially refer to as bags. We prefer the more technical biocontainer term. But now we actually have biocontainers going all the way up to 1,500 litres using identical, very high clarity, very low extractable, low leachable films at the 1,500 litre scale, which go in totes. They're on casters. They're wheelable. You can move them around the plant really quite easily. And think, you save 1,500 litres. That means that you're actually dealing with putting it in real terms is a ton and a half of liquid that you're dealing with in a very robust, very reliable plastic bag system that again gives enormous flexibility to the pharmaceutical plant.
Talk to us briefly about the issue of leachability. I said you brought that up, and that's obviously a concern of customers.
Yeah, obviously, anytime that you have plastics in contact with product, there's a lot of things actually go into the manufacture of plastics, and what you don't want is for those materials that have been incorporated into the plastic at some stage during its manufacturing process, obviously to leach back out of the plastic and come into your product. I can't say that there's going to be none. There will always be some material which comes out, but what we've been able to do is to absolutely minimise the amount of material which will leach out, and to develop analytical techniques. And we've worked very hard to actually develop some leading edge analytics for looking at the things which might potentially be there, so that people who are using these biocontainers have access to a portfolio of analytical methods, allowing them to qualify those films in their process, because they know not only what they're looking for, because we give them chapter and verse on what goes into the biocontainer, but we also give them dedicated analytical methods to look for those very tiny trace quantities of materials in what can often be a very complex mixture, i.e. our customers' products that are stored in the bag. And it's a highly critical area, and we have whole groups of people within our scientific laboratory services and technical support groups who work with people to apply those analytical methods and to help them to actually validate the Allegro systems within their process.
Good. Let's talk a little bit about chromatography columns and sorbents.
Sure.
What's new in those areas?
Yeah, well, the interesting thing about chromatography is that chromatography is maybe a little bit of a bottleneck for some people, because as expression levels are starting to increase, a few years ago 1 gram per litre was kind of like the holy grail for expression. Now we're routinely at 5-7 grams per litre. People are talking about 10 grams per litre, and one or two companies have been recently announced that they'd reached the 20 gram per litre milestone. Now what that means is that for any given fermentation volume, you can have 5, 10, 20 times as much of your target molecule to actually process downstream. Now you can either split it into smaller batches and use your existing equipment. What we wanted to do was to look at the chromatographic media that were out there and say, how can we actually improve these? Ion exchange, for example, has been about, and it's been widely used by the industry. It's the workhorse, but really nobody everybody thinks, well, ion exchange is ion exchange. What can you do to improve that? So what we've done is to say, well, ion exchange, like a lot of chromatographic techniques, is limited by sorbent beads which have small pores in them. The molecule that you're targeting has to diffuse into the pore. It's actually quite a slow process. So what we were able to do was to use our knowledge of how to build microscopic structures to actually make a hollow bead with macropores. So you can think of it as being like a shell, just with lots of big holes in it. Mm-hmm. And what we've done is to fill the shell of each bead with a gel, and we actually put the chemistry in the gel. And what then happens is, instead of having to diffuse into narrow pores, the gel, with all of its active chemistry, actually literally drags the molecules into the gel very quickly. So even for something that people think they're familiar with, ion exchange chromatography, we now have a media which not only can capture things very much faster, 2 to 3 times faster than traditional media, but because we have a hollow bead, all of the inside of that can be used for the target molecule. And so the dynamic binding capacity of the bead is actually 2 or 3 times greater than traditional media as well. Put those two things together, and you have a chromatography media that can give something like 10 times the throughput of traditional media. So that if you have got a process that's gone from 1 gram per litre to, say, 10 grams per litre, you now don't need to invest in new chromatography columns, which are expensive things.
Right.
They're multi-hundreds of thousands of dollars investment. You can actually get maybe up to 10 times the capacity using your existing equipment, and with a sorbent that is in the same price range as traditional sorbents as well, and improving the economics of people's processes through our Process Economics Initiative. This is just one illustration of how we're working to basically to improve the entire process for our pharmaceutical customers.
And obviously everybody these days is cost conscious, so it's important in everything that you do.
Yes, absolutely.
Yeah. How about separations technologies that you've been developing? You mentioned GlaxoSmithKline and some of the work you've done with them.
Yeah, sure. We work obviously with our customers and look at some of the problems that they have, and in the area of recombinant vaccines, a number of people are looking to, rather than having to grow highly pathogenic organisms, is to use genetic engineering to actually express the antigens that are the main part of the vaccine actually on something which is very easy to grow, like E. coli. But if you do that, you need to find a way of actually collecting the bacterial cell walls, and the bacterial cultures are actually quite dense compared with mammalian cell cultures, and very difficult to deal with. So what we were able to do for the vaccine industry was to develop a technology which allowed us to, in one step, collect the bacterial cell walls, to wash them without needing a centrifuge or depth filters or any other type of technology. Just a one-step process that allowed you to then get a concentrate of bacterial cell walls. Now you still then need to solubilize the allergen that you're interested in, but what this PallSep technology allows you to do is then to take the cell wall fragments that you've collected, change the chemistry, solubilize the allergen, put it back through the same piece of equipment, separate off the insoluble component, and the soluble allergen comes through the process. So basically you have a single step, single piece of equipment used in 2 steps of the process that then gives you a very, very clean solution of allergen. And that's already starting to be very popular with vaccine manufacturers, and also with people who actually make things like allergens. If you're being tested for pollen allergies or dust mites or anything like this, Actually, this equipment handles the weirdest and most wonderful types of things. If, for example, you're being tested for dust mites, that actually starts off. Companies actually collect the contents of your vacuum cleaner bag. We can actually run the contents of a vacuum cleaner bag through the PallSep and get a clear liquid come out, which actually has the dust mite allergens in it. It's an odd example, but an illustration of the challenges that Pall sort of rises above the crowd to solve for our customers.
Yeah, as director of marketing, then you have your work cut out for you with a piece of equipment like that.
Yes, that's right. Yes, it's exciting, and I think the main thing is that it shows that no matter what the challenge, we have something in our toolkit which helps to address it.
And my last question will be about rapid microbial methods. I know that you've got some things in the works. What can you tell us right now?
Yeah, sure. Well, we've had the PallCheck system on the market for a while now, which is an ATP-based rapid microbiology system. In fact, it was the first rapid micro system that gained regulatory approval for some of our customers. Now we're looking at the application of PCR-based methods into the rapid microbiology area. We now have some very fast PCR systems available for the food industry. Those are actually launched and they're out there, and we're working to actually use the technology and bring it into the pharmaceutical industry to help people to release product much faster, to look at problems that they might have very much faster, but to be able to do it with a piece of equipment that does not require a team of highly qualified microbiologists to do. So what we really wanted to do was to develop a simple, really foolproof technology that uses single-use technology, but in the laboratory with basically prepackaged devices, prepackaged chemicals, and everything, so that even somebody with the minimum of training can actually do this highly critical task in the shortest amount of time possible. So watch this space. That's coming very soon.
I'm sure you'll let us know.
Oh, of course we will. Yes.
Ian Sellick, thanks so much for talking with us today.
Paul,
really
it's been a pleasure.
It.
Thanks very much.
Same
Thank
here.
you.